The Human Gene Mutation Database collects and presents a comprehensive list of mutations and polymorphisms reported as disease-associated in the published literature. Each mutation is assigned to one of 5 categories based on the level of evidence that is presented for pathogenicity. In this short video, you will learn about the definitions for each of the categories and the evidence that is considered by HGMD curators. Mutations and polymorphisms are entered into HGMD when the authors of published reports show that they are involved in conferring the associated clinical phenotype, or that they exert a direct functional effect. Assessment of pathological authenticity may come from different lines of evidence, including absence in normal controls, novel appearance and subsequent co-segregation of the lesion and disease phenotype through the family pedigree, absence of any other lesion in the gene that could be responsible for the observed clinical phenotype, previous independent occurrence in an unrelated patient, description of a molecular change that is likely to affect protein function, in vitro demonstration of reduced gene expression, mRNA splicing, or activity or protein stability following mutation, demonstration that the mutant protein has the same properties in vitro as its in vivo mutant counterpart, reversal of the pathological phenotype in the patient or cultured cells by gene replacement. Moving in the order of strongest to weakest evidence for pathogenicity, the 5 categories are DM, disease-causing mutations, DM?, likely disease-causing mutations, DFP, disease-associated polymorphisms with supporting functional evidence, DP, disease-associated polymorphisms, and FP, functional polymorphisms. The vast majority of entries in HGMD are categorized as DM, disease-causing mutations that are pathological disease mutations reported to be disease-causing in at least one individual in the literature. An entry is assigned to the less conclusive DM?, likely disease-causing mutations category when it has been reported to be disease-causing in the literature, but where the author has indicated that there may be some degree of doubt or subsequent evidence has come to light in the literature calling the deleterious nature of the variant into question. It is important to note that just because a mutation is categorized as DM disease-causing, it cannot be assumed that the mutation will be disease-causing in all individuals who harbor the mutation due to the complexity of genetics, including variable penetrance and late-onset disease susceptibility. Care must be taken to interpret the likely pathogenicity of a mutation within the genetic context of a particular individual. When a polymorphism, which is generally defined as a variant with a minor allele frequency of greater than 1% in the population being studied, has been reported in the literature to be significantly associated with a disease or phenotype, and a functional effect has been shown, an entry is assigned to the DFP, disease-associated polymorphisms with supporting functional evidence category. If it has been reported to be significantly associated with a disease or phenotype, but no functional effect has been demonstrated, the entry is instead assigned to the DP, disease-associated polymorphism category. Lastly, an entry is assigned to the FP, functional polymorphism category, if it has been reported in the literature to affect the structure, function, or expression of the gene or gene product, but no disease association has been reported yet. HGMD curators frequently review the curated content for entries and make adjustments to their classification in cases where important new information becomes available. All changes in classification are recorded in the variant history. In those rare occasions where an entry has been found to have been erroneously included, or has been subjected to correction in the literature, resulting in the record becoming obsolete, merged, or otherwise invalid, the entry will be reassigned to the R, retired entry category. For questions about classification of individual entries, including suspected errors or omissions, please use the feedback form located within each mutation report, or write to us at bioinformaticssupport@qiagen.com.